Good afternoon, honorable guests, speakers, ladies and gentlemen. And good day to our friends who are now joining us from around the world through the live webinar platform. Welcome to the seminar of the Science Exposition, organized by the Council of the Hong Kong Laureate Forum Limited, which also named as the HKLF. Today's session is particular for life science and medicine, while we will have other sessions for astronomy and mathematical sciences on 13th October. First of all, let me introduce myself. I'm Nick Ting Nong Ai, an undergraduate of the Chinese University of Hong Kong studying medicine. I'm also one of the young scientist applicants for the inaugural Hong Kong Laureate Forum to be held in November 2022. I wish I would be selected and be able to meet with other young scientists and world-renowned scientists in person at the forum next year. The forum aspires to be a world-class academic exchange event to connect the current and next generations of leaders in scientific pursuit, as well as to promote understanding and interest of young generation in Hong Kong and around the world in various disciplines in science and technology. Before the launching of the inaugural forum in 2022, we are embarking on a series of pre-forum events to cater for a variety of audience and further the HKLF's mission of providing a platform for scientists with different expertise from around the world to come together to promote science and enlighten the younger generation in the scientific community. To start off the seminar, may we invite Professor Timothy Tong, Chairman of the HKLF, to say a few words for us. Professor Tong, please.
Good afternoon. It's good to see you again. This is the seminar portion of the Science Exposition, which is the first event that we have organized out of a series of events that will lead up to the inaugural forum that will be presented just over a year from now in November 2022. So today's Science Exposition, we have exhibitions out in the hall, and now this is the seminar series. I want to take this opportunity to thank all the speakers. We have outstanding speakers from the universities in Hong Kong, and I'm delighted to see so many young people in the audience. I think you will find their presentation very stimulating, and I will encourage you to ask questions afterwards. Ask as many questions as you can about the project, about how they became interested in science to begin with. So I want to thank all the speakers, and I wish you a very fruitful seminar series. Thank you very much.
Thank you, Professor Tong. Please be seated. Today, we will have 5 scientists joining us on stage to share their research experience. They are Dr. Loong Hon Nok, Assistant Professor, Department of Chemistry, Hong Kong Baptist University. Dr. Carmen Wong, Assistant Professor, Department of Pathology, the University of Hong Kong. Professor Tom Zhang, Associate Professor, Division of Life Science, the Hong Kong University of Science and Technology. Dr. Bolton Chow, Associate Professor, Department of Rehabilitation Sciences, the Hong Kong Polytechnic University. Last but not least, our moderator of today's panel discussion, Professor Edwin Chen, Professor of School of Life Sciences, the Chinese University of Hong Kong. After each presentation, there will be some time for our scientists to answer 1 or 2 questions. If you have any questions you would like to ask, please click the Q&A window on the Zoom webinar. Then you can submit your questions via the Q&A box. Our scientists will answer selected questions during the session. First, may we invite Dr. Long, Assistant Professor, Department of Chemistry of Hong Kong Baptist University, to present for us. Dr. Long, please.
Okay, hi everyone, distinguished guests and student fellows. It's my honor to be invited by the organizers. And my name is Hong Lok. So today I would like to share with you some of my research projects, right? And that is how basic science can be translated to drug discovery. And it is a study of an early development of agnostic inhibitors for EBV-associated tumors. So what is EBV? The full name is Epstein-Barr virus, okay? And this is a human DNA herpesvirus, which is widespread in the human population, right? And you see the frequency of infection is more than 90% of the population is already infected by this EBV virus. And it was one of the first viruses which was found to be associated with tumor development. And one of the tumor type called nasopharyngeal carcinoma, okay, or NPC, has a very intimate relationship with this virus, and it is 100% associated with EBV. So people in the field tend to believe that EBV is an oncogenic driver for NPC development. And NPC is derived from the normal epithelium in the nasopharynx. So where is the nasopharynx? It's here, as indicated on this diagram. It's just behind the nasal cavity. So anatomically, it's just located almost right in the middle of the head. Okay, so that makes not very accessible for surgical operation. So this is the reason why the first-line treatment for NPC is radiotherapy. And NPC, right, is prevalent in southern China regions, including, of course, Hong Kong, Guangdong, Guangxi, Fujian, etc. And the new cases statistics is indicated in the middle of this line. And similar to other cancer types, the survival rate of late-stage cancer patients is quite poor, right, indicating that the current therapy is not so effective. As I mentioned, the current gold standard NPC treatment is radiotherapy with the adjuvant cystatin-based chemotherapy. But however, none of those agents are either EBV or NPC specific. And more importantly, right, commercially, there's no EBV specific drug. So this is the reason why we attempted to make the anti-EBV drug to treat EBV-associated cancer patients. So this is a proposed model of NPC development. And upon chronic exposure to carcinogen, right, including the nitrosamine, right, from the diet, for example, the salted fish or some other pickled food, right, they contain carcinogen, right? So after chronic exposure of those carcinogens, DNA damage might occur in the normal epithelium in the nasopharynx. Okay, and once chromosome 3p, 9p DNA deletion occur, the EBV will be able to infect those damaged epithelial tissues. Okay, and that will result, right, the EBV latent infection will be result in the clonal expansion of those EBV infected cells. And after that, there will be accumulation of a series of genetic alterations, and ultimately, an aggressive NPC tumor bulk will be formed. So this is the basic biology of NPC development. And in order to escape from the human immune system, the EBV is very smart. It may, Yeah, the NPC is so smart that it prefers, right, to stay on the latent infection, because by doing the latent infection, that can minimize the number and the level of EBV latent proteins going to produce, right? So the immune system cannot recognize the presence of those EBV viral particles. And as you can see, there are different forms of latency, right? Latent 1, 2, and 3. And they're categorized by the presence of the combination of a number of EBV latent protein, including ABNA1, T3, LMP1, and 2, and so on. But the point I want to make here is this table shows the different EBV-associated tumor types, right? But there is one protein called ABNA1. It's present in all the EBV-infected tumor types, okay? So EBV, or ABNA1, right, is the only EBV latent protein which is present in all the EBV-infected cells. And there's a human homolog for this EBV latent protein. So targeting the ABNA1 represents a very safe strategy because that can minimize the chance of having side effect. So what is ABNA1? The full name is called EBV nuclear antigen 1. So from the name you can guess it's expressed in the nucleus. Okay? And the primary function of ABNA1 is to attach the EBV genome or episome, right? It's a circular DNA. So ABNA1 is to attach the EBV DNA with the human chromosome. Or in other words, the function of ABNA1 is responsible for the stable maintenance in the whole cell. And for ABNA1 to function, it has to form a dimer. Okay, so you can see it's composed of 2 monomers. Right? And the dimerization domain maps the C-terminal end of an ABNA1 protein. And this is our drug target. So this slide shows you how we are targeting ABNA1. I used the example, right, called L2P4. It is a peptide, right, containing a peptide we can specifically bind to the ABNA1 protein. And this peptide is conjugated with a small molecule, which is a fluorochrome. So once L2P4 is uptaken into an EBV infected cell, the fluorescent signal will be emitted. Okay? So or in other words, you can visualize the presence or the intake, how L2P4, right, can go into a different subcellular localization in an EBV infected cell. And on the other hand, this anti-EBV agent can disrupt the dimer formation of ABNA1. Right? As I mentioned, if the dimer formation is inhibited, the EBV genome can no longer attach to the human chromosome. And the tumor cell will die at the end. Right? So as you can see, anti-ABNA1 agent has 2 functions, one for imaging, the other for therapeutic intervention. So that represents the concept of theranostics, okay, dual function. But just by the ABNA1 binding peptide itself, it's not sufficient for it to get in the nucleus where ABNA1 is present. Okay, so there's a trick. We've incorporated the nuclear localization signal motif, RRRK, right? We combine it at the C-terminal end of a peptide, which the ABNA1 binding peptide. Okay. And that represents the 2nd generation compound, which is L2P4. So we performed the live imaging using the fluorescent microscopy to study the uptake of our compound. And 2 EBV infected cell lines and the EBV negative cell lines were included in this study. And the blue fluorescent represents the nucleotide, while the red fluorescent represents the signal emitted from L2P4. As you can see in the EBV negative cells, although you can see the red fluorescent, but they're all located in the cytoplasm, but not in the nucleus for all of those EBV negative cells. On the other hand, when you look at the EBV infected cell, the T cell lines, you can see the red fluorescent located in the nucleus. So indicating that the addition of the NLS, the nuclear localization signal, can significantly enhance the nuclear permeability of an anti-Apner-1 agent. And then next, we test the in vivo anti-tumor activity using the nude mice. And we transplanted the tumor subcutaneously, right? It's an NPC EBV positive cell, right? Transplanted into this animal. And then we deliver L2P4 directly into the tumor, right? We do that twice a week. We also measure the average the tumor volume. So in the middle is the result. The x-axis represents the days after treatment, while the y-axis represents the average tumor volume to be measured. Okay, and you look at the red line that is after treatment. So we compare with the solvent control that is without treatment. You can see the tumor volume was significantly reduced after the treatment of L2P4. Okay, so this result indicating that L2P4 has a therapeutic potential. But however, it's not perfect because sorry. Yep. Because when you look at the cellular uptake more carefully for L2P4, there's still a significant amount of fluorescent signal in the cytoplasm, right? So in other words, it's not that specific. So this is the reason why we further incorporated a scintillator to L2P4. And we make the third generation anti-Apner-1 agent, which is called ZRL5P4 or ZP4 in abbreviation. The reason is because there's a zinc finger-like domain present at the N-terminal of the Apner-1 protein, right? And that zinc finger-like protein will function, right, for the self-association that will represent the second dimerization site. Okay, so look at the cellular uptake result of ZRL5P4. But this time the nucleotide is the red fluorescent, while the blue fluorescent represents signals emitted from ZP4. So as you can compare, majority of the blue signal are located in the nuclear. Okay, only very tiny amount signal can be observed in the cytoplasm. Right, so that means the incorporation of this scintillator can significantly enhance the specificity for the Apner-1 protein. But we used a similar animal model to test the in vivo anti-tumor activity of ZP4. So ZP4 could significantly reduce the tumor size. And also, we measure the body weight of the animal. As you can see, there's no significant change of the body weight when compared to the solvent control. So this result indicating that ZP4 is a safe agent for therapeutic intervention, at least for animal use. In addition, we also observe the EBV lytic reactivation after the treatment with ZP4, as reflected by the induction of a number of EBV lytic protein. In addition, the EBV viral particle also induced by our ZP4 drug. And upon the EBV reactivation, the viral particle will be produced in the whole cell. And eventually, this viral particle will burn out from the whole cell, so the whole tumor cell will die eventually. So this is at least one of the mechanisms to explain why the tumor size was reduced after the treatment with ZP4. And besides targeting Apner-1, we also target another EBV-latent protein called LMP1. And LMP1 is a well-known EBV oncoprotein expressed on the cell surface. And the anti-LMP1 and the dual targeting agent, which target both LMP1 and Apner-1, are successfully synthesized. And we recently have published those data in these two publication journals. So in order to commercialize our anti-EBV drug, we have established the HKBU spin-off company called BB Inomac, and it was established in December 2018. And the aim is to make the antiviral agents. So our lead compound, right, had already gone through the drug discovery phase. And the production of the lead compound is almost optimized. So indicating that the CMC, which is a chemistry, manufacturing, and control, we have already have some idea on the CMC. And for the preclinical study, we already performed small-scale preclinical analysis, including the drug efficacy, the drug safety, and pharmacokinetics of the lead compound. So right now, we are doing the large-scale animal preclinical tests, right, to validate for what we observe in the small-scale studies. And our ultimate goal is to put our lead compound to go through and to get the approval of the IND, which is an investigational new drug. Only with that, with the IND approval, we can put our drug into phase 1 trial. If the phase 1 trial is successful, we will license our technology to the big pharmaceutical companies to continue the phase 2 and phase 3 trial and the commercialization process. So thank you very much. I think I want to stop here. And thank you for your attention.
Thank you, Dr. Leung. Please remain on stage for Q&A.
Okay.
Hello, Dr. Leung. We have received a question from online audience.
Okay.
So the question is, does your research also provide some insights as to why nasopharyngeal carcinoma is more prevalent amongst the population in South China? Is it related to our genetic predisposition or our diet?
It's a good question. Actually, there are 3 factors which make the NPC more susceptible. First is genetics, because only the Southern China Chinese, especially for the Cantonese, they're more susceptible somehow to NPC. So genetic must play a role. But research has been done for a number of years, but still we don't know the exact genes that is responsible for that ethical background. And second is diet, right? I think in the 1980s, some Hong Kong research already to show that the salted fish or pickled food are the factor that result in the high incident rate of NPC. So diet is the second factor, right? And the third factor is the EBV, the viral factor. So all 3 factors altogether to play a role for this NPC development.
Thank you, Dr. Leung, for your presentation, your answer. Please be seated.
Okay, thanks a lot.
Thank you. Now, may we invite Dr. Carmen Wong, Assistant Professor, Department of Pathology, the University of Hong Kong, to present for us. Dr. Wong, please.
Very nice music, by the way. So it is my honor to be here to present you my work and to share with you what has been going on in my lab. So I would like to thank the Hong Kong Laureate Forum for their kind invitations, and it's really my honor here. So I worked on cancer. Cancer is the leading cause of death. And it will continue, the incidence will continue to rise. And you can see that in 2030, it is forecasted that there will be more people suffering from cancer. So we have to be ready for this rapid medical demands. So I think all of you have heard of cancer treatments. The most common ones are surgeries, radiations, also chemotherapies, targeted therapies. So all these therapies actually aim to target the cancer cells so then it would induce cancer cell death. Unfortunately, at the same time, it is also inducing the cell death of normal cells, including immune cells. So therefore, there are a lot of adverse toxicity in different cancer therapies. So immunotherapy, unlike the traditional therapy which targeted cancer cells, it's an emerging therapy for cancer treatments. It aims to harness components of the immune system to selectively destroy tumor cells. So it is actually a new paradigm in cancer treatment which aims to reverse the exhaustion of immune cells, to reactivate these immune cells so that they can recognize and kill cancer cells. So let me start by explaining to you how immune cells and cancer cells interact by showing you this cancer immunity cycle. So cancer cells would release antigens into the environment, and that could be picked up by antigen-presenting cells. So these antigen-presenting cells, the classical ones are the dendritic cells, they would travel to the lymph nodes where they meet the T cells. T cells are the major soldiers. So they would present the antigens to the T cells where the T cells would clonally expand, activate it, going back to the tissue site through circulatory system. And then the T cells would recognize more tumor cells with the antigens that they recognize. Then they would elicit their toxicity and kill more cancer cells. In this way, more cancer antigens would be exposed. So you can imagine if, in principle, this cycle is working properly, then all the cancer cells would be eliminated by immune cells. Then there will be no cancer. So here we know that this is not true, and our lab focuses on understand how the cancer cells evade from this immune surveillance. So let me talk about T cells, how T cells are activated. Normal T cells, they receive at least two signals to be activated. The first one is the T cell receptor. It binds to the antigens that is expressed by MHC class molecule presented by antigen-presenting cells, as I mentioned, dendritic cells. And then together with another signal, CD28, which binds to ligand V7 on the antigen-presenting cells. If these two signals are received, then the T cells are activated and they would release cytokines and do their job. However, in order to prevent overactivated T cells or immune system, the T cells actually have self-regulated mechanisms. So they express inhibitory receptors PD1 and CTLA4. So that would actually shut off T cells when they are hyperactivated to prevent autoimmunity. So scientists have discovered that T cells express PD1 and CTLA4. They are the brakes of the T cells. So because of that important discovery, they have received the Nobel Prize a few years back. This is because the discovery has led to the design of antibodies or drugs that block the interactions. So then the T cells can be activated and their activity can be unleashed so they can continuously be activated. So immune checkpoint inhibitors are the inhibitors that block the PD1 and CTLA4. So then it could reactivate the T cells. So these are the most common types of immunotherapies that we will discuss. So apart from the antibodies-based immunotherapies, which involve the blockade of inhibitory receptor on T cells, so there are other antibodies that could activate the activating receptors on T cells. So they are also the common immunotherapies. Apart from antibody-based, there are the adoptive T cell therapy, CAR-T, which involves the isolation of T cells from the patients. And in the lab, we hyperactivated these T cells and also engineered the receptors that enable them to recognize the tumor specifically. And then we culture them in vitro and expand them and reinfuse back to the patients. Another type of immunotherapies involve the direct administration of cytokines to the patients. So those are the old version of immunotherapies. And all these immunotherapies revolve around the principle to redeliver the activated T cells back into the patients. So my lab focuses on liver cancer. Hepatocellular carcinoma is the major type of primary liver cancer. the reason is because it is very lethal and it affects our region. And also, it has a high incidence and its prognosis remained extremely poor because a lot of patients came to the clinic. They are already at stage 3 or stage 4. And unfortunately, we only have very limited treatment options. So it is not until recently immunotherapies, immune checkpoint inhibitors that I mentioned, have been recruited as the standard care of treatments. So PD-1 has just been approved by FDA in about 5 years ago. And then recently, PD-L1 and anti-VEGF combination treatment has shown very good outcome. So all these clinical trials have suggested that immunotherapy, immune checkpoint inhibitors, they actually performed better than conventional targeted therapies. And this is a very impressive study which shows that anti-PD-L1 and anti-VEGF, the combination treatment actually is the first combination or first drug regimen that could beat the conventional targeted therapy that has been used over a decade in the clinic, sorafenib. So while the field is very excited about this promising outcome of immunotherapies, we understand that only a subset of patients responded. So we asked, what happened to the rest of the patients who did not respond to the immunotherapies? So we used preclinical mouse models which resist PD-1. And we performed mass cytometry analysis to study the immune landscape to understand what have been changed after we treated the mice with anti-PD-1. So we performed high-dimensional analysis on the tumor-infiltrating T cells. And we found that actually, when we take a closer look into the T cells, some subsets or clusters are being induced after PD-1 treatment. So what are these T cells? They actually express high level of other T cell exhaustion markers like TIGIT and LAG-3. So I just told you part of the story. Actually, apart from PD-1 and CTLA-4, there are more inhibitory receptors on T cells that we should not neglect. And today I will talk about TIGITs. So we found that after the treatment of PD-1 by flow cytometry analysis, the T cells express more exhaustion markers, TIGIT. And what is TIGIT? So it has a very similar role as PD-1. So when it binds to its ligand, PVR or PVR family, it would also relay signal to shut off T cells. And we found that the ligand is highly expressed in liver cancer tissues. And also it's associated with poor clinical outcome in XCC patients. And when we knock down the gene in cancer cells, we found that the tumor has significant smaller size, accompanied with the restoration of T cell infiltrations. And when we co-culture cancer cells with the T cells, we found that after we knock down the ligand, you can see that the T cell proliferation is increased. And this is actually dependent on CD8 T cells, because when we used an antibody to deplete CD8 T cells, we found that the tumor suppressive effect of knocking down the ligand is gone. So finally, we asked whether we can improve the immunotherapy treatment. So you can see that PD-1 antibody could not suppress the tumor growth, but when we combined with anti-TIGITs, which block the other exhaustion immune checkpoints, then we can see that there's a significant improvement in the outcome of the mice with aggressive HCC. And we want to ask what happened to the T cells in the patients. So we collaborated with clinicians who did clinical trials on PD-1 in HCC patients, and we collected the blood from the responder and non-responders who have undergone nivolumab treatment, which is anti-PD-1. And we found that the non-responders, which mean the patients who did not or received the treatment, actually, their T cells express more TIGITs, further echoing with what we found in our preclinical study. So here in this story, we found that HCC cells have have mechanisms to resist PD-1 treatment because they express the ligands which would interact with another inhibitory receptors on T cells. So that would help the cancer cells to evade from the immune surveillance, and that would help the cancer cells to resist the immunotherapies. And with the co-blockade of PD-1 and TIGITs, we can actually see a better outcome in the aggressive HCC model. So I just told you how T cells and cancer cells interact, but in fact, this is only part of the story. In the microenvironment, actually, there are many more immune cells. Some are the good guys, which I refer to the T cells. Some are the bad guys. So they are the, They are the immunosuppressive cells. So these cells actually could suppress T cells. So I call them bad guys. Okay. So before I go on further, I would like to introduce you to another microenvironmental factor that is important in the microenvironment of liver cancer. So hypoxia actually is a phenomenon that describes oxygen deprivation or a reduction of oxygen level. So it is very common in cancer because when the tumors grow at a rapid pace, it often outpaces the rate of the growth of the blood vessels. So that often leads to insufficient oxygen delivery or blood delivery to some part of the tumors, so creating hypoxic environment. So this is accompanied with the accumulation of this population of cells, myeloid-derived suppressor cells, that I will further describe to you. So actually, how cancer cells adapt to hypoxia, it's mainly through hypoxia-inducible factor, HIF. So this is a transcription factor. Under normal situation, in the presence of oxygen, HIF-1 alpha is hydroxylated at the proline residues, enabling the recognition of one hippolyndro protein that mediates the ubiquitation of HIF and proteasomal degradation of HIF. Okay? So in the presence of oxygen, HIF is always being degraded. Without oxygen, HIF is stabilized because this process cannot take place. So then it dimerizes with HIF-1 beta to initiate the transcription of genes, a wide repertoire of genes important for cancer cell survival. So this, the discovery of this elegant system that regulates oxygen homeostasis has received the Nobel recognitions. And I'm lucky enough to work with Professor Greg Semenza to further his work on HIF-1 alpha. So HIF-1 alpha is actually a central regulator that essentially controls every single hallmark of cancer. And in the next couple of slides, I will tell you how it regulates immunosuppression. So there are many metabolites in the microenvironments, ATP, AMP, and adenosine, but they have very different functions. So ATP actually functions to activate the immune system, whereas AMP and adenosine functions to suppress the immune system. And this is controlled by an enzyme, actal enzyme on the cell membrane, ENTPD2 or CD3901. So it functions to convert ATP into AMP. And we found that this enzyme is actually regulated by HIF. So I told you about MDSCs. So they are actually the myeloid progenitor cells. They're the bad guys in the story. So under normal situation, they would differentiate into macrophage or dendritic cells. But under pathological conditions, they remain undifferentiated and they function to suppress macrophage, dendritic cells, and also T cells. And in our story, we identified that actually hypoxia or AMP could actually accumulate these MDSCs. We first found that under hypoxic condition, this enzyme is being induced dramatically, and this could be abolished when we knock down HIF-1 alpha, the transcription factor responsible for the transcription. And also, we confirmed the direct binding of HIF to the promoter region of this gene. And we performed mass spectrometry analysis, and we confirmed that AMP accumulates in the conditioned medium of cancer cells in the hypoxic condition, and this was abolished when we knock down or inhibit the enzyme ENTPD2. So we asked what happened to the MDSC. So we cultured this MDSC with the metabolite 5' AMP that is induced by hypoxia, and we found that MDSCs accumulate. And importantly, we want to translate this into our clinic. And so we asked, again, we use mouse model to ask whether we can inhibit this hypoxia-induced enzyme, and can it be used in combination with immune checkpoint inhibitors. And we found that this ENTPD2, also CD39L1 inhibitor, could work synergistically with immune checkpoint inhibitors to improve the survival of this, again, aggressive mouse HCC. So in this story, we show that under hypoxic condition, low oxygen, this transcription factor is stabilized, initiated transcription of gene, the CD39L1 or ENTPD2, that would convert extracellular ATP to AMP. This would actually help the tumor to accumulate MDSC, the bad guys that would resist T cells. So apart, due to the time limitations, apart from ENTPD2, we also found that HIF is regulating other chemokines that would recruit MDSCs, as well as lysooxidase, which cross-link the collagen that would enable the recruitment of the myeloid cells. So with that, I would like to thank my team who are also in the audience today. Thank you very much for your support, dedication, and hard work. None of this is possible without my team. And I would like to especially thank David, Vincent, they're Hong Kong students. They have been amazing, and they're doing amazing work in my lab. And I would also like to thank my mentor, Greg Semenza. That was the time when I left his lab about 10 years ago. It was an amazing time when I worked with him and his team. That was the time 2 years ago when he got Nobel Prize. We all the trainees went to his house to celebrate the Nobel Prize, and also for his discovery of hypoxia adaptation system. Thank you.
Thank you, Dr. Wong. Please remain on stage for the Q&A session. We have selected a question from our online audience. And the question is, funding presumably is an important issue for any scientific research. With the onslaught of COVID-19 and the talk of the next virus pandemic being just around the corner. So are you concerned that funding for research on ordinary diseases, such as cancer, be sidelined? How much time do you need to spend on seeking financial support?
Thank you for your question. I was anticipating some kind of scientific questions. Yeah, but this is a great question, by the way, because funding is actually our momentum, because with no funding, then none of this work is possible. So with regards to the question about whether COVID-19 or would all the fundings go to the research in COVID-19, and whether it would affect some disease like cancer. Definitely the pot is there, right? And of course the division, the money is here, the funding is here, but I think it's just about from time to time, right? So funding for COVID, of course, is important, but from time to time, people would remember, like cancer is actually the leading cause. And I would also want to, make some advertisement. Cancer is really an important disease to be studied. So I would further hope that I can attract more fundings by giving this advertisement. But definitely, I think all the diseases have to be funded equally. And I think we all understand that this is important. Yeah.
Right. Yeah, we have one more question.
One more question.
Yeah. Mm-hmm. The question is, do you expect the future of immunotherapy research to be a constant cat and mouse chase due to the discovery of new cell markers, signaling proteins, and the heterogeneous nature of the tumor cells?
I think immunotherapies would, because I think a lot of studies, why it attracts so many basic research scientists to go for understanding the basic mechanism of how immunotherapies work and how cancer resists, is because of the clinical success that we observed. So definitely, I think, I think it will be the trend or the important areas to be studied. So of course, identification of new markers are important. I think not only on new markers, but understanding the mechanisms, how different cell types interact with each other. Yeah.
Yes. Yes, we believe that with all the efforts from the scientists, we can improve the future cancer treatment to benefit more patients in the future. Yeah. Thank you, Dr. Wong, for your presentation.
Thank you.
Please be seated. So now, may we invite Professor Tom Zheng, Associate Professor, Division of Life Science of the Hong Kong University of Science and Technology. Professor Tom, please.
Give it a try. Okay. Well, first of all, I would like to thank the organizer, Hong Kong Laureate Forum, for the invitation to speak here. So my name is Tom, and welcome all. So I understand that today we have quite a mix of audience. So I mean, we have high school students, we have scientists, right? Different types of people. So what I have actually prepped today is kind of a more general lecture about what we usually do. The life of stem cell from young to old. So what we're very interested in our lab is aging. I mean, it's a question that at least personally, I care a lot. I'm getting old, okay? So I will get old as day goes by, right? So the question there is, how do we live a healthy life? But today, what I want to do is going from a stem cell point of view, okay? So the stem cell that we study in the lab is not I think when I talk about stem cells, oftentimes people refer stem cell to the almighty embryonic stem cells, right? I mean, the stem cells that, as you know, the iPS cell, ES cell, these cells that are able to make basically you, right? I mean, from one cell all the way to a human. So the stem cell that I talk about today actually resides in your tissue, okay? So these are so-called the somatic stem cells or adult stem cells, okay? So these stem cells helps you to replenish your tissue throughout the life of your, basically your whole life, okay? So now, maybe before I go into the stem cell, let's talk a little bit about aging, okay? So, I mean, as you all know, we are technology-driven society now. So basically, we have an increase in lifespan as we, progress. Getting to now, we actually, in 2019, the average life expectancy is at 73 years old. Now, the problem becomes we also have an increase in mortality due to the age-related diseases. Okay, so I think Carmen just gave us a lecture about cancer. I mean, this is definitely one of them. Stroke, right? Dementia, all kinds of problems that we have when we get old, right? So the question there becomes, how do we deal with this? I mean, this is a societal problem, right? I mean, we have, on one hand, the quest for immortality, right? Basically, we want to live forever. On the other hand, we have to deal with some of the undesired problems that, eventually, kill us, basically. So now, as it turns out, aging is a risk factor. And many researchers have illustrated that the idea of, turns out aging is malleable. Okay, so what that really means is you can actually reverse aging. Okay, so I will give you some examples of that. So our lab, one of the major focuses is to try to think about the strategy involving slowing aging or to the extreme, reversing aging. And the question there becomes, can we actually decrease mortality? Right, so that's the kind of the big driving force of what my lab likes to achieve in the next decade or so. So now, one of the big questions in the aging field is, if I were to ask you, I think we have many young audience in the room here, right? Of course, you guys, I can tell you guys are younger than me, right? So, but the question there becomes, if I look at, I'll point to our moderator, Edwin, right? Is he older or am I older? So I would say, I don't know. But the question, really is, how do you measure aging? Is our chronological age actually important, right? I will argue that your biological age is more important, okay? So the question there becomes, what is the best way to measure aging? So, of course, we have all these kind of functional assay that we can measure aging. So the Hovax clock, I wouldn't mention too much, but to say this is an epigenetics way, very famous algorithm for measuring age very accurately. So, different kinds of biomarkers, when things gone wrong, right, you have something secreted in your blood, right, you can use that to as a measurement. Frailty, right, you become weak, right, that's a very good telltale sign of aging. Right. So, and of course, cognitive function, I don't have to mention a lot of dementia that you have problems, right? And that also has an indication of, you're getting old. Vitals, right? Heartbeat, all that kind of things. And machine learning, actually, this is the forefront of the AI world. So basically, what people have done is to actually take young and old cells and basically think of Google, okay? I'm sure you're aware that Google is able to recognize different species of cat, right? So think of it the same way. As it turns out, you can also use machine learning to recognize different cells from young to old. Okay, so these are the kind of forefront of some of the methods that people are using to measure age. So now, more important considerations. So, when we measure age, I've already alluded to this a little bit when I say is Edwin older or I'm older. So, as it turns out, our body actually, the tissues within our bodies actually can age differently, right? So, your brain, your heart, your muscle, they can age differently. Okay, so the question there becomes, what should you actually measure, right? These are the kind of questions that, we don't really fully understand. And when you actually take the cells and put it into a culture dish, that also changes the whole thing, right? Because the environment is changing and we're culturing the cells in a very rich nutrient environment, right? So that becomes another obstacle to actually understand aging. So in the lab, what we do is, for the most part, we age the mice for, and we use mouse as a model, and we age the mice for 3 years before we can even start using them for research. So you can see that the kind of work involved and the preparation involved and the kind of design of the experiment involved, you really have to set out the right hypothesis to do this kind of work. Okay, so now, talk a little bit about the path to aging reversal. I think this is probably the most interesting slide that everyone wants to know, right? So now, of course, calorie restriction, exercise, I think these are the things that you hear a lot, right? And it's true, okay? So if you were to do exercise, indeed, it will reduce your apparent age, in your body, in your cells, okay? So, calorie restriction, we eat too much. Don't eat too much, okay? So that's also true, okay? So now, what in the lab, in my lab, what we do, in a way to reverse aging is two things, okay? So today I'm gonna show you one of the story about metabolic reprogramming, but we also do quite a bit this type of experiments called the parabiosis. And this is also the main reason why people say aging is malleable, okay? So the experiment here involved basically a very ancient experiment, okay? So in the 60s, okay, so what people we're trying to study is blood. Basically trying to look at blood types. Okay, so and what they do is they surgically connected the circulating system of the two mice together, and they try to understand the blood chimerism, basically the interconnection of the two mice. So now the technique in around 2000, okay, is being adapted to the aging world, okay, so in which researchers have paired a young mouse to an old mouse. Okay, so the idea there is asking one question: is blood? Are there circulating factors in the blood actually able to rejuvenate an old mouse? Okay, and the answer is yes. So, and as it turns out, this particular system has been proven in, for example, the brain reversal, muscle, kidney, different kinds of organs are able to actually have a beneficial effect by providing some young blood. Okay, so in the lab, what we're very interested in is the kind of circulating factors that are involved within the young blood. Okay, so, and of course, vice versa, we are also very interested in the aging factor, which is something that is being secreted from the old mice. Okay, so you can imagine that these are harmful factors that is actually affecting the young cells. Okay, so, and as it turns out, in the young mice, they do look older, in this particular system. So this basically to say that, when we get old, we do have some aging factor that we need to deal with. And you can imagine that if we can find them, you can eliminate them and you will get younger again. Okay, so now, today we'll talk a little bit more about the metabolic reprogramming. Okay, so, in the lab, what we're very interested in is the skeletal muscle. Okay, and particularly, this is a problem that, every one of us eventually will encounter, so-called the sarcopenia or muscle weakness. I don't have to tell you, your old grandmother, actually, their muscles weak and they would not be able to move around as much as, you do, right? So, and it involves basically the weakness of the muscle, and the muscle, when it gets age, that becomes a problem. And at the heart of this problem, there are these muscle stem cells within there. And the reason why we're particularly interested in using this system to study aging is that, this is a muscle cross section, and you can see these kind of pink dots here. Those are actually the muscle stem cells within your muscle, okay? They do absolutely nothing, okay? So throughout the life of your, whatever that you do, okay? So for the most part, they basically just sit there and do absolutely nothing. When do they have when do they start activating and do their work? When you have an injury, okay? When you have an injury, you can see that the red dots, they're everywhere in the injured muscle, okay? So they help to actually repair the muscle, okay? So now, with that said, this serves as a really good model for stem cell aging. The reason for that is because we know that they're not dividing, they're sitting basically in the muscle throughout the whole life of the animal, the mice that we're using, right? So we can try to decouple the replication problem with this kind of environmental niche, these kind of factors that I was talking about in the blood. How do they actually affecting the stem cells? Okay. So now, I also want to introduce one more concept. Okay, this is so-called the senescence, and senescence is referred to one of the key hallmark of aging. Okay, so as we know it, usually a normal cell, of course, they can do their usual function, and you can consider there's some stress. It can be any kinds of DNA damage, oxidative stress, aging, right? But eventually they become a senescent cells. See a senescent cells as a zombie cell, right? They are alive, okay? So they're alive, they're actually within your body, but they secrete harmful factors, just like what I mentioned earlier, right? In the old mouse, they secrete aging factors, but in the senescent cells, they actually secrete some harmful factors. And eventually, of course, when they do become senescent, they lose their normal function, okay? But they actually contribute to a lot of different kinds of problems: tissue dysfunction, immune cell attraction, chronic inflammation. Okay, so basically what we are very interested in is to see when we get old, or when the mice get old, some of the stem cell actually goes into the so-called senescent state. And the truth is, they do, but only when they get very old. So you can see that this is a proteomic study, meaning we actually look at all the proteins that are available in a cell. Okay, so what we have done here is trying to look at the adult mice, which is around 8 weeks old, the old mice, around 24 months old, and the geriatric, that is about 32 months old. Okay, so the end of life of a mouse is around not more than 36 months old. Okay, so you can consider that the geriatric mice is your 100-year-old person. Okay, so, and as it turns out, you can see the red. These are these are different proteins, and these are all the hallmarks, or basically the proteins that are involved in senescence. So basically what we're saying is the muscle stem cells, when they get old, they actually become a senescent cell. So let me try to, okay, so, and I mean, I've already mentioned that. I don't seem to, okay, great. So I only want to show you a couple slides just for simplicity. And what we found is, I mean, from these kind of studies, we were studying young and studying old mice, we stumbled on one of the RNA binding protein, okay, so called the CPB4, okay, so this protein actually binds to RNA and increase the translation of that particular RNA, meaning whenever you have that, you get more proteins, okay? So now, what happens is during aging, we found that the levels of CPB4 actually goes down. And this is one of the stem cell right here. You can see that the red there, I mean, in the very old stem cell, they don't have CPB4 anymore. So what we believe is, I mean, basically, as the mice get old, right, go into the geriatric, the CPB4 level goes down, and so as the regeneration level, of course. I mean, they don't regenerate very well. So, and what we are able to do is take some old cells, put back the gene, right, put back the gene so you get the CPB4 protein. And what we are able to then do is to take these stem cells and put it back into the mouse, right, through a so-called a transplantation experiment. And with that, I mean, basically, you can see those green looking thing, those are the muscle fibers, okay? So they are able to contribute to the functional regeneration. So suggesting that basically, if we were to take the old muscle, put back the CPB4, we can rejuvenate the stem cells, okay? So, and basically what we have been, heavily working on right now is to try to understand, is this translatable, right? I mean, is this happening in human? That's the kind of work that we do, okay? So, and with that, I just want to acknowledge, I mean, come see us in our beautiful campus. So thank you for your attention.
Thank you, Professor Zheng. Please remain on stage for Q&A session. Professor Zheng, we have selected a question from our online audience.
Okay.
This is actually quite an interesting one.
Sure.
So the question is, do you think that we could produce a therapy that could slow down or even reverse aging once we have cracked the molecular code or mechanism underlying aging?
So the answer is definitely yes. So the reason for that is, I mean, like, this is actually the hardest thing in the Silicon Valley right now. Okay, so if you go to San Francisco, there are in the upwards of 30 different startups dealing with this. And I mean, I think one of the more higher profile one is Google found a company called Calicos, right? So you can see that, I mean, this is going to happen, right? The question there is, when are we gonna crack this code, right? So, but I will say, long story short, if you just go to Crunchbase, this website that talks about startups, and just type aging, I mean, you will see a whole list of companies that are actively doing this. And I would say, I mean, I think for the past 2 decades, right, this is the time. I mean, like 2020, around this time is the kind of the uprising of the aging research, and everyone cares about aging, right? We all want to understand better, can we actually solve aging by solving other age-related diseases?
Thank you, Professor Zheng, for your presentation and answer. Please be seated. Now, may we invite Dr. Bolton Chow, Associate Professor, Department of Rehabilitation Sciences, the Hong Kong Polytechnic University, to present for us. Dr. Chow, please.
So, hello everyone. I would first like to thank the Hong Kong Laureate Forum for the invitation. And it's great to be here because this gives me a chance to share with you something that I feel very excited about. I have a research interest about neuroscience or brain science. So in other words, I study the brain. But I have also a very specific interest in knowing how does the brain make a decision. So it is a relatively new area called decision neuroscience that we study the brain basis of decision making. All right, so it's a relatively young science, and there is some specific interest in the part of the brain called the prefrontal cortex. So the prefrontal cortex is located in an area labeled in red in this picture. So basically, it's the very front part of our brain, and it is located above your eyes. We only have some insight about what this particular part of the brain does over the last 30 years. But before that, the importance of this brain area was overlooked by many scientists. Let me give you an example that happened in the 1950s. There was a very popular surgery called lobotomy that was done in the 1940s or the 1950s. So it is a treatment for severe mental disorders. So it was done in quite a scary way that the medical doctor would take a knife and stick it above your eyes, well, not your eyes, but the patient's eyes, in order to damage this part of the brain called the prefrontal cortex. And it was reported that patients who received this surgery became a bit more quiet. So maybe it's the idea that, well, these patients have some problem. Let's keep them quiet. Let's not talk about the problem. But anyway, it was so popular that even the sister of the United States President, J.F. Kennedy, was also a patient who received this kind of surgery. Her name is called Rosemary Kennedy. So, what have we learned over the 30 years in order to have some insight about what this part of the brain does? This understanding of this part of the brain started in the 80s. One of the examples is about a report about a patient called EVR. So this is EVR's brain. You can see that there's a huge part of the prefrontal cortex that was being damaged. But even with such a huge damage in his brain, he seems to be functioning normally. After the brain damage, he had a few clinical tests. For example, he received an IQ test and he scored quite well with a score of 120 or above. This is even more amazing that he had a memory test and he scored 143, which was an exceptionally high score. Imagine an average person would just score 100. Well, nevertheless, even though he did so well in these tests, he had some problems in his personal life. So he made very poor financial decisions. So after having this brain damage, after he sort of recovered, he went to a business with somebody who was known to be highly unreliable. And because of that business, he went into bankruptcy. And then he also had some marriage problem. He got divorced with his wife. He also changed a few jobs. None of the jobs lasted long. And he even took hours to perform very simple matters. For example, he could spend hours considering where to go for dinner. So, although the clinical test seems to tell that, well, this person is normal, this person is okay, but, well, in his personal life, it was a huge mess. So that indicates that this part of the brain that was damaged in EVR does something very, very important. Subsequently, scientists start to understand a bit more what this part of the brain does. It is indeed related to some decision-making processes. And many studies, including some conducted in our labs, found some consistent findings. For example, if people are asked to choose between an apple and an orange, something happened in the prefrontal cortex. Some people might like the apple more, some people might hate it. Well, it's your personal choice. Different people have different personal preference. But despite this kind of personal preference, we have a part of the prefrontal cortex, which is called the ventromedial prefrontal cortex, that reflects how much that we like the apple and how much that we like the orange. So basically, this part of the brain is above your nose, quite at the center of the prefrontal cortex. So what happens to this part of the brain is that if you like the apple a lot, this part of the brain will become highly active. But if you hate the apple, this part of the brain will be inactive. And the same would be true when you're looking at the orange. Not only between food choices, that also can be applied when you're making financial decisions, or even your own preference to some piece of artwork, whether you like it or not. This can be told by your activity in this part of the brain called ventromedial prefrontal cortex. So basically, these many studies showed the first time that there is something physically there in our brain that tells about our preference. And this is very important for our decision making. So let me tell you some other facts about the prefrontal cortex. So if we look at different brains, the brain of humans, the brain of monkeys, the brain of the dogs and cats at your home, we do have a very large prefrontal cortex. So it's basically very large. So the black area labeled in these figures is the prefrontal cortex. Some scientists even argue that some other animals might not even have a prefrontal cortex, for example, mice and rats. Some people think that, well, it doesn't exist in these animals. Another interesting fact is that since we were born, our brain keeps developing. And the prefrontal cortex is one of the last areas that gets fully mature. It takes 25 years for the prefrontal cortex to be fully mature. So I guess, well, some of you might not even have a fully mature prefrontal cortex yet, but this might be a good news. You have still got a lot of potentials in the future. The question or the problem is now that, well, even though I told you that the prefrontal cortex is important for decision making, but other animals that do not have a prefrontal cortex, or younger people with a premature prefrontal cortex, can still make a decision. So the critical question is that, well, what does the prefrontal cortex in our brain do that makes us a human? That makes us making so many different types of intellectual choices. So why do we need the prefrontal cortex for decision making? So I start to have some observation about, well, what is unique about our choices. We are quite good at making choices with complex information. For example, if tonight you are thinking where you should go for a dinner, you might switch on your mobile phone app And there would be lots of different restaurants around this area. So we are in the Xiqu Centre, which is around here. And you can see that, well, there are plenty of different restaurants. And it's quite a difficult job to find a restaurant that you would like to go. But you can still do it reasonably well. You might spend 10 minutes, 15 minutes to do that. So we're good at processing complex information. But in order to test that in the laboratory, we ran an experiment with my student, Michael Wu. So we asked volunteers to come to our laboratory with an empty stomach. So they were very hungry when they visited us. And we gave them snacks to choose from, like between 16 snacks. And we asked them to do this kind of task repeatedly. So what they needed to do is just to pick the favorite snack. And we tried to observe how they did that. To cut the story short, we realized that one part of the prefrontal cortex is particularly useful when we are making these kind of choices with complex information. And this is the part called the dorsolateral prefrontal cortex. What it does is to help us to filter important choice information. Because you know that even though in your mobile phone apps there are plenty of options, hundreds of restaurants, most of the restaurants are irrelevant to you because, well, for example, you're a vegetarian, you just don't eat meat, so you just ignore the rest. Or you know that some restaurants are not good at all, you just ignore them. The prefrontal cortex or the dorsolateral prefrontal cortex have this specific function for us to help us to filter important choice information. Another important observation about human decision-making is that we are quite good at making choices with long-term consequences. For example, you might be considering after your study what kind of job you would like to take. And this kind of decision does really have an important consequence for at least the next few years of your life. Or another very important decision is to select a partner, which might even have a huge consequence for your lifetime. And basically, we're so good at doing that because that we can imagine what will happen in the future if we make this decision. We can also try and integrate different aspects of information in order to guide this kind of decision. So in order to address how that happens in the brain, again, we try to set up some experimental task. We invite volunteers to our lab and make some choices. And in this experiment, we designed a task that mimics choices with long-term consequences. Again, sorry that I have to skip the details, but at the same time, when people are playing this kind of game, we ask them to stay inside an MRI scanner. So it's a scanner that takes picture of the brain, but it does not only help us to understand brain structure, but we can set it in a way that we can detect brain activity. The interesting finding is that we found that a part of the brain in the prefrontal cortex called the frontal pole is specifically involved if we're making these choices with long-term consequences. One exciting observation about that is that, well, although I told you that other animals, they still have a prefrontal cortex, although they might be a bit smaller. But this part of the brain called frontal pole is unique in human being. You can't find a similar frontal pole in cats and dogs or monkeys. Okay? So this is really something quite unique in us, and that might explain why that we are so good and we have been evolved in a way to allow us to make these long-term choices. Not only that we try to understand what's happening in the brain, we also try to develop an AI algorithm, artificial intelligence algorithm, that chooses like a human, first of all. So it behaves like a human. This AI, I have to emphasize that it knows nothing about the human brain. It has no knowledge about the human brain. But what we did is to make a comparison between the AI and the frontal pole. And the critical thing is that we realize that these two systems, the AI and the frontal pole, they share similar computational processes. So the AI basically does something that is a bit like the human brain. The reason why I would like to bring this up is because there's great parallels between AI and neuroscience, the science of biological intelligence, that I would say. The human brain is made up of simple processing units called neurons, and they all are connected to each other to form a neuronal network. In the same way, the electronic brain, or the AI, was developed based on this observation about the human brain. So in the AI, nowadays you have artificial neural networks that is composed of artificial neurons, and they're connected to each other in order to do different types of jobs. It can be used for making a decision, it can be used for recognizing faces, and so on. A few years back, you might have heard of a news that, the best board game player was defeated by the AI called AlphaGo. So they were playing this board game called Go, and this is the human player, the Korean player called with the surname Lee, and this is AlphaGo. And people start to ask, well, are we not as good as the AI? Does the AI make better decisions than us? I'm not going to tell you the answer, but I just want to tell you some facts. Well, let's compare the human brain and AlphaGo. The human brain contains 100 trillion neurons. Okay, it's a huge amount. And AlphaGo contains 0.7 million artificial neurons. And there are such a number of connections in the human brain and in AlphaGo. So you can see that there is a huge difference in the complexity between the human brain and artificial intelligence. However, we consume way less energy. To keep the human brain going, you just need 20 volt. So basically, you just need rice to keep you going. With AlphaGo, it consumes way more energy to keep it going. AlphaGo is composed of a number of computer servers, but even for just one server, you need 500 volt. And with the human brain, it can perform a lot of different functions, not just playing board game, but, for example, to meet your friends, to talk, and to recognize things. AlphaGo is just for playing board game only. So I argue that the future is that we would like to know why that happened in the human brain. Why is our human brain so efficient? And how can we try to apply these kind of knowledge in order to help us to understand various things? For example, the development of artificial intelligence. So I think that's the end of my talk. I would like to thank different parties, people in my lab, my collaborators, my funders, and for your attendance. Thank you.
Thank you, Dr. Chow. Please remain on stage for the Q&A session. There is a question from the online audience. The audience is interested in your next experiment. What's your future research direction?
That's a good question. There are a number of them. The easy one would be to understand cognitive functions, such as how good you make a decision, how good is your memory, and so on, based on brain data. Can we just capture a picture of the brain in order to forecast how well you're doing these things? Well, of course, this is the next goal, but it would be a very difficult goal, and that would require the collaboration between many different parties.
Thank you, Dr. Chow, for your presentation and answer. Please be seated. Thank you all for the presentation. I believe you all should now have a better understanding on these projects that you have visited just now at the exhibition. For our audience currently staying outside Hong Kong, we have not forgotten about you. There will be highlight clips of the research showcase available on the HKLF website later on, so please stay tuned. It's now the time for our panel discussion. We will be learning more about having research in Hong Kong and the latest research capacity and opportunities. We also welcome questions from you anytime during the session. Again, please click the Q&A window on the Zoom webinar. Then you can submit your questions via the Q&A box. Our scientists will answer selected questions. We are very honored today to have Professor Edwin Chan, Professor of School of Life Sciences of the Chinese University of Hong Kong, to be our moderator. Professor Chan, please. And let's welcome our 4 speakers to be on stage again.
Thank you very much for the organizer to invite me to join this very exciting and meaningful and challenging job. So I'm your moderator today, and my name is Edwin Chan. So in the past hour or so, we have listened to the presentations from different speakers and our outstanding young scientists in Hong Kong. So what we heard were the most exciting and forefront research activities going on here in our city. And what I could say is I saw actually different scientific approaches, and also saw different research interests from the work that presented by the presenters. And more interestingly, I saw different research capacities. For example, I saw very good drug development program. I saw very good approaches to disease pathological studies like in cancer field. I see how we can stop aging or even reverse aging soon. And lastly, or most importantly to me, how we could make decision at the right time. Like now, I should please be stopping to talk more than to let the scientist to talk to you and interact with you. So to begin with, I would like to really, I want to say something. Science is a very huge field. And sometimes you may say, oh, I'm a scientist because science picked me. Okay, not I'm choosing science. So I'm not a scientist because science didn't pick me. Is it really true? To me, I don't think this is the only way to explain why we are all now doing a scientific research job. So the first question I would like to ask all the scientists on stage now here is why you chose science as a career in the first place? And why are you still doing it even after like so many years of ups and downs? You're still doing whatever you do. Quite a boring job every day in the lab, writing or in the office writing grants and so on. So maybe any one of you can say a few words on this.
Maybe I can start first. I think during high school, I already enjoyed science very a lot. I mean, I knew I liked the subject of chemistry, biology, and physics, and maths. I enjoy a little bit of history, but other than that, I don't enjoy other subjects at all. So obviously, I'm born to like science somehow.
The science really picked you then.
I picked science. Science picked me. A perfect match.
Okay, what a good match.
Yeah, somehow it works. Somehow I still survive here. Though it's not easy, but because I like science, I can tell you the working life in lab, it's a whole day, 7 days a week. If you don't enjoy working in lab, that will be very tough. I think all of us have that kind of experience. So I think the interest is my driving force for me to sustain in this area. So yeah, this is my experience.
Yeah, great. Thanks, Hao.
You have some.
Well,
yeah.
maybe I'll say a few words. I'm sure many of you like video games. So do I. So I love video games when I was in, primary schools, secondary schools. And one thing that I did back then is I like to crack the games. Okay, so I like to cheat to try to how to break this game, right? So one time I actually stumbled on one of the TV show talking about the genetic code. Okay, so these genetic codes, I'm sure by now you know the genome, A, T, G, C, there are a lot of actually hidden messages in there. And I fell in love with it. Basically, what I really like is to try to find out how do I crack the genomes. Okay, so that's me.
Okay, similar to what Tom has said, my Game Boy was with me all the time when I was young. So I have many hobbies. So I'm not like this typical nerdy scientist. So I just noticed that I am a very inquisitive child. I was a child, and I was very curious. Till now, I'm also curious in everything. So my parents always say that I ask too much. So I keep asking them, why, how, why is the sky blue? I start to ask questions and try to observe small things. I still recall the time when I was a high school student. I had this science class. So I did not care too much about the scores at that time. So I know in Hong Kong, it's difficult not to only work for curiosity, but science is something about curiosity, right? And I'm not good at everything. It's during the journey, you identify what you're good at, right? So I recall the time when I was a student in high school. I asked my teacher, science teacher, can I stay a little bit behind and ask you some questions? Before that, I actually drafted a list of questions. Yeah, a list of questions beyond what he just taught, because I was just interested to know more beyond the syllabus. Yeah, so that's how I discovered my interest in science, and I don't think any other subjects could fulfill my insatiable desire to something that I want to know. Yeah, so science is one of them. So I hope you will start to ask questions and try to observe the details instead of just being stressed by the scores. Yeah, so this is how I discovered myself in science adventure. Yeah.
Bolton?
There's something I don't quite agree with you, Edwin, because you said, well, why do we still stay in this career, given that it is boring? But I think that, on the stage, none of us would think it's boring. That's the kind of thing that I'm pretty sure. Somehow there are boring elements, but in general, I think most of us enjoy our work. And just like Carmen, just like Tom, I also play a lot of video games. I hope that won't stop teachers in the future bringing them to this kind of event because, professors saying that they play video games. I guess that, I myself, I just would go into those things that I really like, and just like Hong Lok, that I would not be convinced to do things that I don't like. And that's why I also I did very badly when I went to secondary school, because I was asked to study a lot of different subjects I didn't like Chinese history, right? But I do like Chinese history nowadays. But I think the kind of thing that keep me going is to really identify something that I really love to do. It can, for some people, it can be playing video games, and just go for it. Go be a pro gamer. But if you like science, then go for it, be a scientist. Then as long as you stay focused on your interests, if you can keep going without giving up, then you can do well.
Yeah, I do hope that Bolton will have your device further developed, and so we will have a test somehow in the future when the kids go to your lab, put on the helmet, and then you just give them, okay, doctor, lawyer, scientist, and your ventral frontal cortex would just light up. And that would be the future scientist. I hope that would be some kind of development. But anyway, I think only the 5 of us are sitting on stage, but there are like thousands of scientists in this world, and I'm sure that their own stories are all different. But I think listen to your heart is important. I mean, why you're still here, I mean, in this room now, but without like going out for dinner, as what Bolton has mentioned, did give you some part of the answer to science. So I got one question from the web, asking about a very interesting and ever-developing field, which is called cross-disciplinary research. And what do you think about cross-disciplinary research? So you all talked about your own research, but I do know that you all collaborate with different scientists in different fields. Do you think interdisciplinary research would go further than just you being alone as a sole investigator in your research program?
Well, maybe I'll say something first. So, I mean, interdisciplinary research is something that is, I mean, it's been around for a little while and it will be here to stay. Okay, so the reason is very simple. I mean, I mentioned about machine learning, right? So you can imagine that as biologists, we're no good at computer science, right? So how are you going to utilize the state-of-the-art machine learning to do your, kind of image classification, right? I mean, that is what Google does best, right? So, I mean, many of these kind of issues started to show up when we starting to get into the so-called the big data era, right? So, I mean, we have a lot of data, and I think one of the very, clear message is, I mean, you need to, choose one good area, right? But you also need to know a little bit of other areas so that you can actually communicate with the other experts, right? So, I mean, you don't need to know the inner details of the other field, but you definitely need to, I mean, talk, right? I mean, you have to have basic knowledge to talk about it, right? So, or to get people, I want to do a machine learning in these cells, right? I mean, at least you need to know, I mean, the little where to get help to do that kind of work, right? That's my view to it. Mm-hmm.
If you see my CV, I'm from the Department of Chemistry in HKBU. But I can tell you, myself, I'm a biologist. Okay, so that is very strange. How come a biologist working in a chemistry department? Because of collaboration, right? As I tell you in my talk, I'm making anti-cancer drug, right? But my role is to identify the drug target. It's not exactly, I'm not the one who's making the drug. It's a chemist who's making a drug, okay? Because I don't know how to synthesize those small molecules. So it's really the effort of the multidisciplinary result by combining the field of biology and chemistry. Because I know I've been working on cancer biology for more than 20 years. I'm very familiar with the cell lines. I'm very familiar with the animals, and I know the drug targets are there, but I don't know how to make the drug. So this is the reason why I need someone who is interested to make a drug from the Department of Chemistry. I think my chemistry collaborator always say one phrase. He said, 1 + 1 not equals to 2. It's more. It's really, especially when the time I'm submitting my manuscript to the journal. I don't think there's an expert who has expertise in the 2 areas. The good thing is they don't have much comment, right? Either they don't understand the biology part of my manuscript, or they don't understand the biology part. So it's really making something very new, very novel. So this is how I see it. It's really a trend of the modern research.
Yeah, Great, thanks, Hong.
I think, as I said, nobody knows everything. So it is very important that we collaborate, and also I think definitely interdisciplinary research is important. Doing science is like playing basketball game, right? You cannot score by yourself. You need some team members to help you score. Some are better at playing center, some are better playing forward, some are better at wing position. So doing science is something like that. So we need to help out each other in order to score and reach our goals. And we all have our strengths and weaknesses, and we need to learn from each other. It's a continuous way of learning. Yeah.
Yep. I think, at least in my area, is surely an interdisciplinary area because, well, first of all, you need to know about behavior. You need to know what is happening in the brain. Or if you have a little bit of interest in AI like I do, well, you also need to know a little bit of that as well. I think previous speakers already have mentioned, nobody is good at everything. That's why we need to do cross-disciplinary research and collaborate with people. But I also think that, well, in order to do that, sometimes it can be quite tricky, but at least you need to have a little bit of knowledge about what other people are doing. You don't really need to be an expert, but at least you need to have a bit of knowledge in order to let yourself to communicate with these people.
Yeah, thank you so much. So one fun part about collaborative research or interdisciplinary research to me is you go and make friends from time to time. I still remember I just go to a seminar, and at the end, I introduce myself to the speaker saying, oh, I'm from my university, and I would like to really collaborate with you on certain area. And then we became very, very close collaborators for years to come. So one fun part is you're going to make friends along your way if you decide to go on to do interdisciplinary research. And in the end, just like what Hong said, 1 + 1 never just equals to 2. It's more than 2. And that's the important and fun part of science. So because of my interest of time, then I should go for the last comment or questions. So my last question to all of you would be, how are you going to encourage our young scientists or younger generation here, and also on the web, to pursue science? Other than science is fun, because science picking me, any other things that you would like to tell them?
I think it's about finding your interest and then stick to that. And then that's it. It can, as I said, it can be science, it can be anything, but you just need to find something that you really feel excited about.
So in my view, resilience. Okay, so you really, I know you guys are working very hard right now, okay, and you're spending a lot of time studying your DSE, okay, so, but that's just the beginning, right? So you really, when you get into undergrad, that's still the beginning, okay? So, for people like us, we actually spent 15 to 20 years studying, right? Doing PhD and then postdoctoral work, right? Before you get to actually run your own lab and do your science, right? So in my view, resilience will be something very important. Okay, so you gotta, work towards your goal. And the only way to do that, just like what Bolton said, interest. Drive by your interest and you will always get there.
So I think I echo with what Tom has said. I think perseverance, it's very important if you want to do scientific research. And also you need to have a heart to face failure. Yeah, because it is a marathon. Life is a marathon. Studying is also a marathon. So do not give up by only one failure, because you will have different encounters and you just grow through these failures. So I think if you have this heart, then you're suitable for doing research. And I just want to round up my discussion by saying, shoot for the moon, you may land among the stars. Yeah.
I really agree with what Carmen mentioned, and yeah, lots of failure in doing experiments. I always tell my people, my students, you do a lab work, you expect 90% of your time are just failure. So maybe only the 10% thing going to work, and you really have to have the interest and curiosity. I think curiosity is very important. You want to know more, then you don't mind the failure. I think you can come across that. So you just ask yourself, how big is your curiosity? Is it big enough to overcome all the failures? Right? We all have up and down, right, during our career. But I do looking forward to have more young scientists to enter the field, because I really have big problem recruiting the research scientists in my lab. Really, I'm looking more and more talented young people, right, to join our party. Right. Thank you very much.
Thank you. So finding what you love to do, and do not fear to fail along your way, and keep your curiosity going along your journey in science, I think would be what bring us up on the stage here today. And I hope that in years to come, many of you would choose science as a career, and then maybe 10 years later, one of you down here will be standing up here, maybe too soon, 10 years, 20 years from now, and sharing with your juniors, okay, your own journey in science. And I think with that, I wish all the best to the 4 young and rising stars scientists, the best in your research. And I hope you all enjoyed this evening or this afternoon's presentation and have an enjoyable evening. Thank you very much.
Thank you all. This is a very enlightening and thought-provoking discussion. I hope all of you have benefited a lot from the speakers today. So time flies, and we've come to the end of the seminar. Once again, we'd like to express our gratitude to all the scientists and their teams for the time and effort put in this seminar and exhibition. We'd also like to thank you for joining us, not only those physically sitting here in the seminar hall, but also many of you are joining from around the world via the online platform. Thank you all once again, and wish you have a pleasant day. Thank you.